My name is Alex Vodenlich. I'm the president and CEO of Gentel Biosciences, a biotechnology located here in Fitchburg, Wisconsin, at the Fitchburg Technology Campus. Gentel has been around for about 9 years, although the early years were doing something totally different than we're doing now. We've reorganized and repositioned the company to focus on developing and manufacturing and selling protein microarrays. Protein microarrays are little devices that are made that allow the testing of multiple analytes on a little chip. And so what we do here is we manufacture them, we print the test materials onto the chip, and then we sell these to researchers who want to measure these various proteins that the test is designed for. And so we're working to develop tools to provide clinicians and researchers and people studying these proteins and what they do to give them the reliable tools in a very cost-effective, affordable way. The protein microarray products we manufacture here really start with a foundation which is a glass substrate, so it's a microscope slide, that has a very specialized coating. And the importance of this coating is that the proteins that or the test that you spot on that, it has to be retained onto the surface. It has to have very low background so that there's maximum sensitivity. After that, it goes into a different part of our manufacturing process. And this is where there's a very fancy machine that actually spots proteins or antibodies in an array on the surface of this slide. And so you take that and essentially package it up and sell it to a researcher. And so what a researcher will do is we'll open up the package, have a slide, put it in a special cartridge or cassette, add the patient sample, and then go and put it into a scanning instrument. And the scanning instrument that we've developed is a low-cost scanner called the GR8. Here you've got 8 slides times 16 tests that you just simply close, hit the scan button, and 4 minutes later you've got data that looks like this. You have the glass slide that has 16 different or 16 arrays printed on it, and each array has a number of tests essentially printed. And so, for instance, this set of 4 spots at the top, you're testing for one protein. This second set of spots, you're testing for another protein. This third set of spots, you're testing for a third protein. And the novelty here is that in most tests in a clinic, you measure one thing at a time, right? Here we can measure up to hundreds of things at a time. And so what we're looking in a patient's sera is for several or tens of 20s of proteins changing with a disease state or with a treatment or a therapy to see how those signatures are changing. So it's really on the way to personalized medicine, where you combine this with information about a person's genetic makeup as well.